Και περνάμε στο τελευταίο session της πρώτης ημέρας του σχολείου. Αν είναι συνδεδεμένοι οι ομιλητές από το εξωτερικό, Νίκο. Οπότε μάλλον θα μιλήσω λίγο αγγλικά για να τους καλωσορίσουμε. We welcome our speakers from abroad in this session of pharma industry research and development in immune-mediated diseases. We, with Panos, consider pharma industry as a partner in clinical and basic science research. And with this concept is the second time that we invite people from the research and development part of the pharma industries to take part in our in the field of rheumatology. Maybe autoimmunity they have a long presence, but it's only for the last 3 or 4 years that we have contact with them. And it's Lilly and AstraZeneca. So Panos is, I think, better familiar with AstraZeneca because they are our school. So with this, I would like to welcome the first speaker, which is Elizabeth Riedl. Elizabeth is a senior medical director, therapeutic area leader in immunology in Lilly International. And we welcome Elizabeth and...
Very good. So let me share my screen. Okay. Yes, good evening. Thank you for the kind introduction and thank you for having me. And it's a great pleasure to speak about, for the next 20 minutes or so, about Lilly's commitment. As you said, I'm the therapeutic area leader for our international business unit for immunology. I'm a dermatologist and a dermatopathologist by training, and exciting to be here today. So before I go a little bit more into our pipeline in immunology, I would like to share some global fast facts about Lilly. As you know, Lilly is a global company. It's located in Indianapolis in Indiana, and actually approximately 8,000 of the 34,000 employees at Lilly are working in research and development. And we do have research conducted in 55 countries, and we do have facilities dedicated to research and development in 7 countries. So this is a commitment that the company has, and if we go now specifically to our focus areas in immunology, it is really 3 aspects that I would like to mention and which I would like to talk about in the next couple of minutes. We do have assets that are really like the foundational success of our immunology portfolio, which is Taltz, an IL-17A antibody with indications in psoriasis, psoriatic arthritis, and ankylosing spondylitis, and Olumiant, a JAK1 and 2 inhibitor in RA and atopic dermatitis. And we also have in this area, in phase 3 development, antibodies against IL-13 in atopic dermatitis and IL-23 in inflammatory bowel disease. But another focus with some of our earlier assets is immune resolution. And these are really emerging assets that where I want to highlight an IL-2 conjugate. And I will speak a little bit about this. And the third pillar is our approach with innate immunity assets. And here I want to focus on an CXCL1-2 receptor ligand antibody that we're developing currently. So if here you see an overview of our immunology pipeline, and as you can see, it is a quite broad pipeline that we have from phase 1 to phase 3, and this is really something that is a strong foundation. It is also reflecting the direction that Lilly has been taken over the last years immunology. And what I want to highlight, especially in this presentation, is are three of the assets that we are developing in phase 1 and phase 2, which is a small oral small molecule in direct against IL-17A, and the IL-2 conjugate I just mentioned, and the CR1/2 ligand monoclonal antibody. So as I said before, we do have with TALs a very efficacious and successful biologic that is approved in psoriasis, psoriatic arthritis, and ankylosing spondylitis, which is targeting IL-17A. But still, looking at psoriasis and other immune-mediated diseases where IL-17 plays a key role, there is still an opportunity for oral medications. And this was really why we set out a couple of years ago and tried to find a way to design an oral medication that is actually targeting IL-17. And this has been done and now tested in a phase 1 trials. And what differs with this small molecule is that it has the potential to bind IL-17A intracellularly. The small molecule wedges between the IL-17A homodimers and thus prevents the receptor binding. And this has been tested in a mouse model, and it has actually here tested versus the TALs, so versus the IL-17A antibody. And as you can see here, that with increasing doses, we achieved comparing efficacy and time to onset of clinical score improvement. So this is obviously a very exciting asset that we have, and an oral medication targeting IL-17 has obviously other potential indications beyond psoriasis and psoriatic arthritis, and it's something where we really exciting to develop this and explore various indications where IL-17 is known to play a role in the pathogenesis. Another approach to autoimmunity and to try to find new targets and ways of treatment for autoimmune diseases is not by suppressing T effector cells, but actually by trying to restore homeostasis and to target T regulatory cells. And this is what we tried with a new asset that we have. It's an IL-2 conjugate that has been developed together with a partner, Nectar. That's also where the name of this compound is coming from. And this is a pegylated IL-2, which binds to the high affinity IL-2 receptor on regulatory T cells, with the idea to expand the regulatory T cells without having an effect on T effector cells. And also here, I want to share with you some phase 1 data that were performed as a proof of mechanism. You see here the results from a single and multiple ascending dose studies in healthy volunteers. And what you can see here on the upper panel is the expansion of regulatory cells with the IL-2 conjugate. And in the lower panel, you see that the effect on effector cells is actually quite small. And you'll see that here with both the single dose and the multiple ascending dose study, we found about a 15 to 17-fold increase in the induction of T regulatory cells. This was obviously exciting, and the question is then always, how does this translate into clinical efficacy? And this is an early study that was done in lupus patients with a skin and looking at the skin score effect of this compound. These were really preliminary studies, but what we found is that there is an effect on these patients that have some level of skin involvement with ascending doses of this conjugate, while you see here that within the 28 days that this study was performed, there was no effect with placebo. So very exciting data. And lupus is one indication that is currently in phase 2 clinical studies with this pegylated IL-2. But there are other indications that are of high interest and that we are studying. Inflammatory bowel disease, but also psoriasis and atopic dermatitis are currently investigated in phase 1 studies. And finally, I would like to introduce to you another antibody that we are currently developing. It's a pan-CXCR1/2 ligand antibody. It is an humanized IgG antibody that has been engineered to recognize a common epitope on the CXC chemokine targets. It has a high affinity for the neutralization of these targets, but does not interfere with other critical host defense mechanism. So this is an asset that is currently studied in a skin disease, in hidradenitis suppurativa, which is a chronic common inflammatory disease, which is primarily located in apocrine-rich areas of the body. And the hallmark of this disease is really a suppurative neutrophil-rich inflammation that starts at the hair follicle and has then a very chronic and recidivating scarring and fibrosing course. It is associated with other comorbidities, including cardiovascular disease, diabetes, and inflammatory arthritis. So this is an interesting indication for such an antibody to study, and we are very excited to see how these studies are going. And finally, I want to take a few moments also to speak about our commitment in immunology that goes beyond actually drug development. And this is maybe not so broadly known, but I think it's worth mentioning that, of course, a big part of our commitment is not only to develop drugs, but also really to generate data and disseminate data related to therapeutic areas of focus and for our treatments. But it is also to support independent research through investigator-initiated trials for scientific questions and areas that we do not follow as a company. And we also have strong partnerships and sponsorships across the globe with international and national societies, with research societies and fellowship programs. We do have an international grant program for academic centers, which also includes fellowship programs in both rheumatology and dermatology. And we are supporting medical education and run medical education programs, but also support independent medical education. So I think that's one important asset of how we show our commitment to immunology. And I hope that this brief overview was helpful for you. And I wish you, a successful meeting. And I hope very much that very soon this can be face-to-face again for all attendees. Thank you for your attention.
Thank you, Elizabeth, for this nice overview. It was focused, and we are very keen of hearing thus focused presentation from the pharma industry. Any questions for Elizabeth for these new areas of research in the field of autoimmunity? So, my co-chair, Panos Verginis, who is an associate professor in medical school, has a question.
So thank you very much for the very nice presentation. I have two quick questions. The first is regarding the IL-2. And from your results, it seems that the higher doses of IL-2 give better results. And I would like to ask, how is this compared to the literature that shows that lower doses in IL-2 in graft versus host disease and also SLE mediates the expansion of the regulatory T cells and remission? And the second one is, I saw in your pipeline that the phase 2 has some PD-1 agonists there, and I was wondering if there is anything that you would like to share on this. Thank you.
Yeah, thank you for the question. So to the first one, it is really related to the compound that has been developed, so to this pegylated IL-2 construct, and I cannot go into real detail here, but it has a its binding capacity and receptor affinity is related to the IL-2 construct. For the PD-1 agonist, yeah, that's an exciting approach because it goes in the same direction where targeting a checkpoint molecule actually uses the other arm, so to say, for resolution of inflammation. And so far, PD-1 antibodies are only known from or have been used in tumor immunity in tumor therapy. So this is a different approach and something that's also developed in currently in a lupus trial.
Thank you very much. Any other questions?
May I ask Elizabeth for the CXCR inhibitor? This is a macromolecule, correct? It's a monoclonal antibody.
This is an IgG antibody, yeah.
Okay, so do you plan to target other diseases that are mostly related to innate immunity, like these autoinflammatory syndromes?
Yeah, usually at this stage, I mean, this is now the first indication where we went into the clinical development, but the approach usually is to go as broad as possible, and but at the moment, hidradenitis is the lead indication for this antibody.
Because we have so many other diseases like familiar Mediterranean fever or the autoinflammatory syndromes that they are so common, at least in our areas, that we have many therapeutic, gaps.
Yeah, I agree.
Okay, so if there are no other questions, I would like to thank Elizabeth for this very focused presentation, and Panos is going to.
So thank you.
Thank you.
We will continue.
Thank you.
And immune-mediated diseases. Δεν ακούστηκε τίποτα. Okay, so should I repeat? Okay. So it's our pleasure to introduce you the next speaker, Dr. Brohawn, and I say again that I'm not sure whether I pronounce the name correctly. So he's a Senior Director in Translational Science and Experimental Medicine of AstraZeneca, and his talk entitled the type 1 interferon signature, its development and application in SLE and immune-mediated diseases.
Thank you very much for the introduction. Just want to check and make sure you guys can all hear me clearly. Is that correct?
Yes.
And by the way, the pronunciation of the last name was spot on, much better actually than is typically done in the United States. So,
okay, great.
Very well done. So today I'm going to switch gears perhaps a little bit and talk about type 1 interferon gene signature and then regulation of the type 1 interferon pathway as a therapeutic approach. My colleague Hussein will speak after and kind of give some insight into the clinical results from impacting the type 1 interferon pathway. But first, I wanted to go through how we leveraged our biomarker development efforts to not only identify or enable the drug development process of type 1 interferon inhibition, but also to look at life cycle management and disease selection, sort of to the question there that at the end of the previous talk. So let me advance slides here. So the development of a type 1 interferon gene signature or a type 1 interferon transcript signature was key to enabling the development of clinical programs inhibiting the type 1 interferon pathway. And it was a complex set of preclinical experiments that led us to the identification of this signature and then the further development and technical validation. And what you have here in the Venn diagram is simply a depiction of the experimental process that we undertook. So if you look here at the light blue circle on the outside, that's indicated 807 type 1 interferon inducible probes or genes. These were identified by a simple stimulation experiment of type 1 interferon stimulating whole blood to identify what transcripts were type 1 interferon activated. The right-hand aspect of this image, the green circle, represents the 161 probes that were transcripts that were induced by SLE patient serum and then neutralized by the inhibitors of this pathway. In this instance, I have sifalimumab listed. We also have anifrolumab in the pipeline, which has recently been approved in the United States and Japan under the trade name Safnello. This experiment was fairly straightforward, but quite insightful. We took normal healthy donor PBMCs here, stimulated them with the serum from SLE patient sera, or from SLE patients, and then inhibited those. So either pre-incubated with the inhibitors or with a control. And we identified here that there was 161 of these transcripts that were both induced and then able to be neutralized by an inhibitor. And the reason we did this was to assess the pharmacodynamics or the on-target effect of our molecules so that we could potentially identify these markers in patients. And then finally, the left blue circle here is shows the number of transcripts that were actually upregulated in the whole blood of SLE patients. So it doesn't do us a whole lot of good to develop markers for indicative of disease or on-target effect if we can't identify them in the patient segment that we're targeting. Ultimately, the overlap of these different signatures led us to a panel of markers that we identified as pharmacodynamic markers, helping us further refine dosing and make dosing decisions in the future and understand pharmacokinetics, PK, and PD relationships. And if we look at the distribution of this particular signature, we identify a very unique and sustained feature of the type 1 interferon transcript signature, and that's this bimodal distribution. So oftentimes in biomarker development, we have continuous distributions, and therefore assessing a threshold or a cutoff becomes quite challenging, and we're reduced to empirical assessments like median, quartile, and tertile-based assessments. Here with a bimodal distribution, we have the ability to separate these groups out into those patients that are clearly type 1 interferon elevated and those that are not type 1 interferon elevated. And we set the threshold for this differentiation in the trough of this bimodal distribution. And while I don't show it here, I can tell you that we have evaluated this signature with multiple different methods, including transcriptomics by next generation sequencing, microarray, qPCR, as well as surrogates of this signature, cytokine-based, and we observed this same bimodal distribution across multiple different technological platforms. On the right-hand side, you simply see here what would be type 1 interferon high or type 1 interferon low, and the distribution of these two subgroups. And you can see here that the medians or the means of these groups is quite significantly different, and that there are very few patients that sort of lie in this trough indicated by the distribution plot on the left. And so what do patients with an increased type 1 interferon signature look like from a clinical perspective? And this is data from our phase 2 studies and published in ACR a number of years ago, where we saw strong associations with those folks with elevated type 1 interferon signature and higher levels of oral corticosteroid use, higher levels of standard serology measures, and then higher levels of disease activity as well. So elevated CLASI, an indicator of skin activity, and then the SLEDAI-2K also being over 10 had a strong association with type 1 interferon response or type 1 interferon status. And so what does a type 1 interferon gene signature positive patient look like? In summary, they're more clinically and serologically active patients. So I mentioned that we identified this signature to demonstrate pharmacodynamic effect or on treatment effect of the compound or the drug itself. And what I'm showing here is a pharmacodynamic plot of the changes in type 1 interferon gene signature over time. This is from an earlier study. This is from the MUSE study, demonstrating that we have sustained suppression or sustained on-target effect of anifrolumab, starting as early as 12 weeks and sustained throughout the entire dosing period. This particular image doesn't demonstrate it, but we also saw dose-dependent response or dose-dependent return to the baseline levels of type 1 interferon expression on a dose-dependent manner following removal of study medication. So telling you that this is a reversible phenomenon and that it is impacted by continuous treatment. However, I've shown you earlier that dichotomous breakdown, and one of the observations made early in the development of type 1 interferon inhibition, an observation that was made is that the type 1 interferon patients that have elevated type 1 interferon signature at baseline are those that are more likely to respond, i.e. their disease is driven by type 1 interferon. And so with that recognition, in early clinical studies, we utilized a research use only or an internal assay to understand and assess the type 1 interferon status. We then selected a partner to develop a clinical trial assay, what is known in a number of cases as kind of a homebrew or LDT type assay to assess our phase 2 population. And furthermore, we've developed this assay into a commercial ready kit, and that commercial ready kit or investigational use only assay is what we deployed in our phase 3 clinical studies to stratify our patients into either type 1 interferon high or type 1 interferon low. And then this is enabling potential regulatory submission should we note that we need to enrich for patients with type 1 interferon signature ultimately on the market. And what would this assessment look like to a physician? It's fairly straightforward. It's a 2.5 ml blood draw. It's a standard 2-step qPCR process, isolation of RNA, generation of data, and there's a built-in algorithm that would simply kick out type 1 interferon test elevated or not. Fairly standard turnaround times should be in line with the serological assessments that would be standardly performed on a lupus patient. So how else are we leveraging, assuming that patients respond regardless of type 1 interferon status, or that we see response in type 1 interferon signature low patients in addition to signature high patients, what other value can the type 1 interferon signature have in terms of refining patient populations? And this is a slide pulled from a book that was written a number of years ago, indicating that this bimodal distribution that I described earlier is sustained across a number of additional indications. So here in green, we have lupus, in blue, lupus nephritis, in red is Sjogren's syndrome, and in yellow is rheumatoid arthritis. Now, to orient you to this slide, the left-hand side of this slide depicts patients with a higher type 1 interferon. The right of the dashed line represents lower levels of type 1 interferon signature. And we've also included some healthy donors here for a reference. But in summary, what you can see here is that there is a subset of patients across all of these disease types that show a clear, distinct overexpression of type 1 interferon signature. And that while that prevalence may vary from quite high in lupus nephritis to perhaps quite low in rheumatoid arthritis, it does represent a unique opportunity to identify these patients via a blood-based type 1 interferon signature, where potentially type 1 interferon inhibition therapy could be beneficial. And so two such diseases in which we've identified clear biological rationale for potential type 1 interferon inhibition are scleroderma, or systemic sclerosis, and myositis, which I'll get to in the next slide. And very clearly, what you observe in this middle image is a clear upregulation again of type 1 interferon signature in a subset of scleroderma patients. In the dot plot or the distribution plot below that, you can see that the level of type 1 interferon activity in the whole blood is similar in magnitude to that we observe in SLE. And again, it demonstrates this bimodal distribution. Perhaps even more telling in the upper right-hand quadrant is that we have correlative overexpression of type 1 interferon or correlative roles of type 1 interferon signaling, both at the site of disease, or in this case, skin, on the y-axis, and in the periphery, blood on the x-axis. So not only do we have activity of type 1 interferon at the disease site, indicating its biological relevance, we can detect that level of activity in a broad sense in the periphery. And then finally, on the bottom right-hand side here, you can see an image from the anifrolumab phase 1 study, which was actually performed in scleroderma, where we saw strong suppression of the type 1 interferon signature despite a dose escalation in the skin of affected patients. And so the phase 1 study was run in scleroderma. We saw strong suppression, sustained suppression of that type 1 interferon signature in these patients. And then also in myositis, there's a strong rationale for the role of type 1 interferon, particularly in dermatomyositis and a subset of polymyositis patients. In the upper left-hand quadrant here, what you see is relative to normal, a clear overexpression of the type 1 interferon signature in the whole blood of dermatomyositis and a subset of polymyositis patients. And then to the right-hand side, we also see this same upregulation again at the disease site. While we can assess the levels of type 1 interferon activity at the disease site by biopsy only, having a surrogate to be able to assess in the blood is key for this biomarker to be deployed clinically. But it is comforting, and from a biological rationale perspective, to observe this type 1 interferon dysregulation at the disease site. In the bottom left-hand side, you can also see that IHC images of these myositis muscle biopsies indicate a clear infiltration of PDCs, which are a key producer of type 1 interferons. And similarly to what we observed in SLE, it's not all patients that have type 1 interferon activation. It's a subset of these dermatomyositis and polymyositis patients that exhibit this clear overexpression, which again can be identified through a blood-based assessment. And if you look at the upper right-hand quadrant, we got some confidence from a phase 1 study with the sifalimumab or interferon alpha blocking monoclonal antibody that we see a reduction in this type 1 interferon signature when we introduce an inhibitor. We were able to capture longitudinal biopsies, and we saw a strong suppression not only in interferon alpha type 1 interferon signature itself, but also in other immune-mediated markers or transcripts. And finally, we saw early associations of the levels of type 1 interferon and response. We saw better MMT8 improvements in those patients with a stronger reduction in their type 1 interferon signaling, giving us some confidence that perhaps type 1 interferon plays a key role in this disease. And so this is how leveraging early biomarker work from in a drug development setting can help us not only enable the primary indication, but also enable identification of additional lifecycle management opportunities or additional therapeutic areas where we may be able to provide significant benefit to patients. And with that, I will stop and welcome any questions that you may have.
Thank you very much for the nice presentation. Any questions from the audience? Okay, let me start again. So if I understood correctly, the interferon gene signature was about 160 or something genes, correct?
So there's multiple. Maybe perhaps I can go back to the slide and explain. In fact, what's interferon inducible broadly is more like 800 and 800 plus transcripts that are differentially expressed. What we identified here in terms of the gene signature, those are probes that are induced and then neutralized by type 1 interferon inhibition. So yes, 161 in that case.
Okay, so the question is, in the patients that were the signature low, did you observe, so did you measure significant interferon levels in the serum? So this is the first question.
Yeah, so initially, actually, the type 1 interferon signature, transcript signature, was developed due to lack of reagents for detecting type 1 interferons themselves in the serum. So actual detection of cytokines was challenged by availability of reagents, antibodies, essentially, for capture, which is why the transcript signature was developed as a surrogate or a downstream effect thereof. But we do see differential regulation, so we do see elevated levels of type 1 interferon cytokine now in the context of elevated type 1 interferon signaling. Although the magnitude of this and the dynamic range of the levels of expression are quite different. So this assay appears to be a little more sensitive to changes in type 1 interferon concentrations.
So for that reason, I was trying to think why a patient could have type 1 interferon but does not engage the signature.
Yeah, so, and that's a great question. So signaling could be impacted by any number of different cases, but we don't typically see high levels of type 1 interferon cytokines themselves without signaling, at least not in the context of outside of therapeutic interference with the type 1 interferon pathway. The anifrolumab antibody itself blocks the receptor. Therefore, we see clear lack of signaling, but we also see an accumulation of the cytokine, which then can't signal in the extracellular space.
Okay, thank you.
I have a simple clinical question. What is the percentage of the patients that they have this high interferon signature? It's 5%, 10%, or
it's an outstanding question. It depends on the disease. In lupus, we've observed in our clinical studies approximately 80% of patients that have a clear overexpression of the type 1 interferon signature. In other diseases, that prevalence varies substantially, but it is a significant proportion of the SLE patients that exhibit this type 1 interferon signature.
And the specificity for response was high?
Yeah, so another great question. So specificity for response is a little bit more difficult to really characterize. So you'll be aware that the SRI-4 and the BICLA are difficult endpoints and heterogeneous. And if you look at the data here in terms of PD suppression, it's clear and sustained with not a lot of variability. So it's very difficult to say. We do have patients, for example, that have strong suppression of type 1 interferon, but would not be categorized as responders. While there is an enrichment for those with stronger PD suppression and response, it's not a direct relationship.
It's the same biomarker that you have in the study of anifrolumab, or it's a different one?
It's the same one.
Okay, thank you.
So just a last question. Since you said 80% of the SLE patients, I was wondering how specific to type 1 interferon are those genes? I mean, do you know if any other cytokines can also regulate their expression and tested positive?
Yeah, so we have looked pretty substantially at other sources. I mean, even type 2 interferons in terms of inducing these genes. And we think that they're fairly specific. In addition, the fact that we're inhibiting the type 1 interferon receptor and we drive specificity from that regard. So we do feel that they're largely type 1 interferon specific, but these are transcript signatures, so they may not be perfect. In terms of selecting the transcripts themselves as well, we selected those with the largest magnitude of overexpression, as well as those that were most readily neutralized by inhibition of all type 1 interferon signaling by the receptor. So there's additional confidence provided by that sort of selection process.
Thank you very much.
Thank you.
So if there are no other questions, we thank Zach Brohawn for the presentation. And we move to the last speaker, who will be Hussein Al-Mossawi, and I hope that I pronounced it correctly. He is coming from AstraZeneca International, and the topic of his presentation will be the clinical benefit of inhibiting type 1 interferon biology in SLE.
Thank you. Thank you for the organizers for giving us this chance to present the work. I follow on from Zach, who's given us a great introduction into type 1 interferon biology and the signature development. I'm a rheumatologist by background. I work at AstraZeneca in the clinical development side of the organization. And what I want to do today is walk you through some of the phase 3 clinical data from the anifrolumab studies in systemic lupus erythematosus. It's fair to say this is an area of high unmet need. And until a few months ago, with the approval of anifrolumab Safnello in the United States, there have been very few approvals in this space and lots of failures. So it's an area that we've invested heavily on because we believe that this drug can make a difference to patients. And I guess the key question that we try to address with this clinical program is this interferon signature that we observe just a correlation of an ongoing inflammatory response, or is it indeed a driver of pathogenesis? And we believe it's a driver of pathogenesis, and therefore inhibiting it should provide clinical benefit. I'm going to try to move my slides here. So I'm going to give you the, it's an extensive clinical development program in systemic, in lupus. What I'm going to focus on are the TULIP studies, which were the 2 phase 3 studies in this disease. I'm just going to get rid of my picture because it's getting in the way. And so I'm going to go through a slightly different order. I'll do the TULIP 2 study first, which was a positive study. And as a lot of you who followed this field may know, that the TULIP 1 study was not a positive study, and we will try and address some of the reasons behind that, and some of the lessons we learned in terms of developing in this disease and bringing this drug to patients. So overall, these were fairly similar trial designs. TULIP 1 had 2 dosing arms. TULIP 2 was a simpler 1-to-1 randomization. Both of these were 52-week studies. And those of you who look after these patients with lupus know that one of the biggest unmet needs is the glucocorticoid use with its associated side effects long term. So one of the things that we really try to address as part of these studies is seeing if we can bring down the glucocorticoid dose with a mandatory glucocorticoid taper, but still maintain patients and maintain that clinical response. And I'll show you some of that data in a moment. And just the 2 programs had different endpoints, and it's fair to say BICLA was used as the endpoint for TULIP 2, as per the sort of BILAG definition, whereas TULIP 1 used SRI-4. And I just want to address this upfront, because I think the key difference between these 2 endpoints, for those of you who treat patients with lupus, you'll recognize this, is that the SRI-4 is an all or nothing measurement. And in some sense, a partial improvement doesn't register in a SRI-4 response, whereas we believe that BICLA is a more is an endpoint that allows us not only to see full resolution, but a partial improvement, which is clinically meaningful for patients. So this is the overview of the TULIP 2 study. This was, as I said, a randomized controlled study, and I'll go through some of those details in the next few slides. Here's the randomization that I showed earlier with a long-term extension. And here's the requirements for a BICLA response, which is a reduction of BILAG scores in the different domains, but no worsening as well. And that was quite key to the response. And no worsening in disease activity is measured by SLEDAI or patient global assessment. And no use of restricted medications beyond those allowed in the protocol. So these were a typical SLE population, as you might imagine, as defined by the ACR criteria. Patients had to have moderate to severe SLE with a SLEDAI 2K of greater than 6. They had to have positive serology. And we allowed, because we believe this is important, we allowed background use of immunosuppressive therapy, such as antimalarials, hydroxychloroquine, and mycophenolate. We excluded from this study patients with lupus nephritis. We're doing a separate program in lupus nephritis because the endpoints there are different with regards to the kidney, and we excluded neuropsychiatric lupus. So these are the characteristics of the patients in the study. As you might imagine, in lupus population, these are predominantly females. And this is what Zach was saying earlier, 83% of those who went into the study, both in the active and placebo arms, had a high interferon gene signature score. The study included patients with Asian genetic background and Afro-Caribbean genetic background. And as you can see, about half the patients in the study were taking doses of glucocorticoid greater than 10 milligrams per day. And this is the primary endpoint. So it was clear that there was a response, and a statistically significant response, in the anifrolumab group, as defined by the BICLA. And what was interesting is that we began to see improvement in this next slide 8 weeks into the study. So the response to anifrolumab, the response to blocking type 1 interferon biology seemed to be rapid and sustained across the year, and clearly differentiating from placebo. And bear in mind, this is on top of standard of care. The key secondary endpoints for the study included a sustained glucocorticoid reduction, which I'll show you more simply in a moment, and an improvement in the CLASI activity response. This is the response of a skin-specific measure, but also annualized flare rates. This is, after all, a relapsing, remitting disease with flares. And here you can see that 50% of the patients in the anifrolumab study were able to achieve a sustained dose reduction of the steroids below 7.5 milligrams a day. So what happened in TULIP-1? And I think it's important to be upfront and share the lessons we learned in trying to develop anifrolumab. There are two key, and I'll go through these results in a moment, but there are two key lessons learned, I think, from our perspective. Lupus is a heterogeneous disease, and it's a relapsing, remitting disease. Often patients who come into the disease to qualify and to get in with the disease activity measures need to have, are in the midst of a flare. And there's a natural regression to the mean, which is why in not just our studies, but in a lot of lupus studies, you see a relatively high placebo rate, because you need to have a high disease activity index to get into the study. So these are patients in the middle of a flare who will regress to the mean naturally. The two other things we learned are you need to pick an endpoint that's sensitive to a partial improvement, as you'll see in a moment. And definitions of what were restricted medications were perhaps a little bit too stringent in this study, and I'll show you some of that data, and I'll let you make that decision. So in this study, there were two doses investigated, 150 milligrams and 300 milligrams. There were 2:1:2 randomization. I'm going to focus on the 300 milligram versus placebo data for the remainder of this talk. You can see the stratification criteria were similar. There was a mandatory glucocorticoid taper. And back to this, here the SRI-4 was the primary endpoint, and it was a greater or equal to than a 4-point reduction in the SLEDAI-2K score from baseline, and no new severe disease activity, and no deterioration from baseline of the patient global. But if you remember, you need a complete resolution of a manifestation to change the SLEDAI-2K score. So if your skin improves, or for example, if the double-stranded DNA comes down by half, that doesn't register, even though that's a clinically meaningful improvement. So this is just revisiting those response criteria. And these were the data from the results of that TULIP-1 study. And as you can see, this study failed to show a clear difference between anifrolumab and placebo for the primary endpoint. So we went back, and the team spent a lot of time to try and understand why that was. We were convinced of the biology. And there were two things that we looked at. And the first was pre-specified restricted medication rules.